Good morning. So I'm going to show you the results we recently obtained regarding a functional screening we have performed in the lab to identify microRNAs which induce cardiac regeneration. So in order to identify microRNAs which potentially increase cardiomyocyte proliferation, we performed this screening, an unbiased screen, for cardiomyocyte proliferation using a library of all annotated human microRNA mimics. So basically what we have done was to array the microRNA mimics on 96 well plates and transfect cardiomyocytes isolated from newborn rats, and 72 hours later look at different proliferation markers. We looked at alpha-actinine as a marker for cardiomyocytes, and then two different markers, Ki67, which generally labels cells in the cell cycle, and incorporation of EDU, which is a nucleotide analog incorporated during DNA synthesis. So these are images of our negative control, a C. elegans microRNA. On this side, you have the original image where you can see cardiomyocytes stained in green, and then cells where the nucleus is labeled in red. So if you have here a cardiomyocyte which the nucleus is labeled in red, it means it incorporated EDU, and therefore it's a proliferating cardiomyocyte. You can also appreciate, since these are primary cultures, that we have cells which do not label for alpha-actinine, which are more likely fibroblasts and endothelial cells. On this side, you have the segmentation analysis, and here you can see the cardiomyocytes stained in green, and if they have a yellow nucleus, it means that it's a proliferating cardiomyocyte. And using this approach, we could determine that in this control situation, we have 12.5% of the neonatal cardiomyocytes proliferating. Now, these are the results of the screen, and you can immediately see that we have a significant number of microRNAs which strongly increase cardiomyocyte proliferation. So we have about 200 microRNAs which increase proliferation by more than twofold. And we also have the opposite effect. We also have a significant number of microRNAs which strongly decrease cardiomyocyte proliferation, so about 300 microRNAs. So this becomes more clear if I show you some representative images of microRNAs increasing cardiomyocyte proliferation. Here we have two top hits of the screen, and you can immediately see that the number of cardiomyocytes which are now positive for the proliferation marker is dramatically increased. This is also clear here in the image analysis, and you can see that now we have about 50% of the cardiomyocytes proliferating, coming from a control situation where we had 12.5%. As I said, we also have the opposite effect. We also identified microRNAs which strongly decrease cardiomyocyte proliferation. These are just two examples, and I should mention that these microRNAs decrease cardiomyocyte proliferation without affecting cardiomyocyte viability. So you cannot find green cells with a yellow nucleus. So one thing that we immediately look at was to see if families of microRNAs, so microRNAs which share the same common seed sequence, would induce the same phenotype. And indeed, we find this, and here I'm showing you, for examples, two microRNA families where all the members of these families increase cardiomyocyte proliferation, and two different microRNA families where all the members of these families strongly decrease cardiomyocyte proliferation. I should also mention that the miR15 family has in the meantime been shown by Eric Olsen's lab to be involved in the regulation of cardiomyocyte proliferation. So we had 200 microRNAs, human microRNAs, which strongly increase the cardiomyocyte proliferation of red cells. And we now wanted to restrict this number and to find microRNAs which are likely to work in human cells. This is why we started with the human library. So what we decided to do was to retest these 200 microRNAs in mouse cardiomyocytes, which have a low proliferating capacity. And if we now identify microRNAs which can commonly increase the proliferation, we hope that they will also work in human cells. So here we retested the 204 microRNAs, and we were able to identify 40 microRNAs which increase proliferation of both rat cardiomyocytes and mouse cardiomyocytes by more than twofold. So you can argue at this point that what I'm showing you is an increase in terms of DNA synthesis and not true cardiomyocyte proliferation. To answer to this question, what we have done is to look at different proliferation markers. So in the screen, we use Ki67 in the EDU incorporation, and now we looked for these 40 microRNAs in two different proliferation markers, namely phosphorylation of histone H3 in serine 10, which labels cells in late G2 or undergoing mitosis, and the presence of mid bodies, which is a structure which is formed when the cell is undergoing cytokinesis. So here I'm showing you some images. For example, in this case, we transfected the cells with microRNA, which strongly increases cardiomyocyte proliferation coming from our screen. And you can immediately see that now the number of cells which are positive for phosphate H3 is much increased in comparison with the control. And this we have done for the 40 microRNAs we identified, and we have been able to quantify this both in rat and mouse cells. The same is also true for the presence of mid bodies. Again, with this top hit from the screen, we do see an increased number of cells which have this mid body structure, which appears during cytokinesis. And again, we could confirm this for the 40 microRNAs coming from the screen. Even more clear than looking at the proliferation markers is counting the number of cells in the well. If indeed you are inducing proliferation, then you should see an increase in the number of cells. And we look at this here for the results of the screen, and you can immediately see that for the microRNAs, which increase the positivity for the proliferation markers, we also have an increase on the number of the cells in the well. This is done 3 days after transfection. Now, if you look at 6 days after transfection of the microRNA, this becomes even more clear. This is the cells transfected with the control microRNA. These are the cells transfected with 4 hits of the screen. You can immediately see that now we have the plate full of cardiomyocytes. One important point that I should mention is that these microRNAs that we identified, which increase cardiomyocyte proliferation, do not induce hypertrophy. And this is important if we want to take them for a therapeutic approach. So the next point that we looked at was to try to identify targets of these microRNAs. And for this, we used the following approach. So first of all, we transfect mouse cardiomyocytes with 2 top hits of the screen, 2 different microRNAs, and we did a transcriptomic analysis by deep sequence to identify messenger RNAs which are downregulated by these microRNAs. In this case, we could put a cutoff value of 1.5-fold. We identified about 700 messenger RNAs which are downregulated by these microRNAs, which are likely targets of these microRNAs. So, but now we have this number of messenger RNAs. How do we see which one of these messenger RNAs are responsible for the phenotype in terms of proliferation? So what we did was to do a targeted siRNA screen, where we specifically knock down each of these genes and look at, using our proliferation assay, if they do induce the same phenotype. And we were able to identify that depletion of 45 of these 600 genes do increase cardiomyocyte proliferation by more than 2-fold. And these are the results of this targeted siRNA screen and the genes which increase the proliferation by more than 2-fold. And what I want to highlight from this slide is if you look at these results, although all of these siRNAs do induce proliferation of cardiomyocytes, none reaches the level which is induced in terms of proliferation by the microRNAs. And this is in agreement what we know for the function of microRNAs. They don't act on a single target, they act on multiple targets, which will give a common phenotype and a stronger phenotype than depleting each of the single targets. So I show you that in vitro we can induce cardiomyocyte proliferation using microRNAs, but can we also do this in vivo? So for this, the first approach that we used was to inject intracardiacally in newborn rat hearts the microRNAs, the synthetic microRNAs, and then look at the proliferation 4 days later. If you focus on these images, which is our proliferation marker EDU, you can see that the number of positive cells for EDU strongly increases when compared with the heart of the animals injected with the control. So maybe you have already seen these images around, and you can immediately see that, and now I can explain to you what these images are. So these are hearts of the animals which were injected with the 2 microRNAs coming from the screen, and this is the heart of the animal injected with the control. You can immediately see that the hearts of these animals are increased compared with the heart of the control animal, and although it's not very obvious from these images, as I showed you before, this is due to proliferation and not from hypertrophy. And we can say that the cells which are proliferating are indeed cardiomyocytes, as I can show you here in these representative confocal microscopy images, where you see that the nucleus of the cells which are positive for the proliferation marker are indeed cardiomyocytes. Then we wanted to induce long-term expression of these microRNAs, and for this we used AAV vectors, and we injected AAV vectors encoding these microRNAs, IP, and look at their effect in newborn mice, and then we look at their effect 12 days later. you can immediately see here in these arts that the animals injected with an AAV encoding for the microRNAs are much bigger than the control arts, and this again is not due to induction of hypertrophy, as we can show by determination of the cardiomyocyte cross-sectional area, but it's indeed due to proliferation, as I can show you here, for positivity for the phosphostone H3. And I should also mention, for example, in these representative images, that not only we see that the cardiomyocytes are positive for phosphostone H3, we also see a disorganization of the sarcomeric skeleton, which would likely be necessary for the proliferation of these cells. So I show you that the microRNAs also work in vivo. The most important question is then, would they have therapeutic value? If they induce proliferation, can they also have a beneficial effects? And for this, what we did was to use a mouse model of myocardial infarction. We injected in this border zone of the infarction the AAV encoding for the microRNAs, and then we performed the echo study 12 days, 30 days, and 60 days after the injection of the microRNAs and myocardial infarction. And we looked at several cardiac parameters. Here I'm showing the results for ejection fraction, fractional shortening, and left ventricular anterior wall thickness. And as you can see, for example, if we look in more detail to ejection fraction, we have in the animals injected with the 2 microRNAs a complete restoration of these cardiac function parameters to almost the level of non-infarcted animals, and this becomes even more clear if you look at the results 60 days after myocardial infarction. If you now look at the size of the myocardial infarction in the animals injected with the control, you can see that we have big infarctions where when we injected the animals with with AAV encoding for the microRNAs, the infarction area is clearly reduced, as you can also see by our quantification. And to end my presentation, I also wanted to show you that we know that in part this effect is due to an increase, to an effect of these microRNAs in inducing proliferation after myocardial infarction, as you can see by these representative images of the border zone of the infarction area, where we do find cardiomyocytes which are positive for the proliferation marker EDU. So to summarize, I've shown you that we have performed high-content fluorescence microscopy-based high-throughput screening to identify microRNAs which might regulate cardiomyocyte proliferation. And using this approach, we identified 40 microRNAs which increase proliferation of both rat and mouse cardiomyocytes. And I've also shown you that we have selected 2 microRNAs for in vivo study, and that both of them are very effective in driving cardiomyocyte proliferation, and more importantly, that the delivery of these microRNAs after myocardial infarction not only restores cardiac function, but also induces myocardial regeneration. And finally, I would like to acknowledge the people which contributed to this work. First of all, my mentor, Mauro Giacca, people from the lab which contributed to the work, and also Miguel Mano from the HTS facility to perform the screen. And thank you for your attention.